Thank you, Ram, and it's a great opportunity to talk to you here in the room and remotely, and those who are watching the video. So three different ways. Yes, I did. I am. So yeah, just a brief introduction. So I used to work in Hungary. I did my PhD there. Started as a biochemist, and then started working more with signaling networks. I'm going to present. And 9 years ago, I got a fellowship, and I moved to Norwich, where basically I was working in the Earlham and then the Quadram Institute to establish kind of my independent work around gut systems biology, and the group that works currently with cell and cell microbe interactions, with an interest in precision medicine and personalized microbial therapies. So the group is quite multidisciplinary. We are generating or working with publicly available on different omics data sets. We do different data integration methods with them, and then computational analysis. And then either with collaborators or with our own group, we are able to do different validations with the predictions. And the central topic in our group is signaling networks. And then we are interested about their relevance in host microbe interactions, in the regulation of autophagy, and the disease aspect we are quite interested in the last 6-7 years is inflammatory bowel disease. I'm going to mention that. And experimentally, we are working with organoids, which I also going to cover briefly. And we have a couple of nice clinical collaborators on these. So basically, the talk will have 4 parts. The first part will be like a general introduction to network biology, and then network medicine in relation to inflammatory bowel disease. And then I would like to present briefly 3 use cases, 3 different types of network approaches. And if time permits, I'm going to mention the new BRC organoid facility, which basically officially started last week. And I'm co-leading. So I guess many of you have already seen some molecular interaction networks. These are really nice, very simple, abstract visualizations of complex processes. You have nodes and edges there, and then you can add different annotations or attributes to these, and that makes it really versatile, what you can do. So a node can be a protein, a gene, a metabolite, and you can add where it is actually located, what is the topological role in a signaling or a metabolic process. And then for the edges, you can add the type of interaction. Is it an enzymatic reaction? Is it a binding? Is it a metabolic reaction? You can have a direction, activatory, inhibitory effect, and these type of things that can actually help to make it a very nice model system. In case of signaling networks or signaling processes, the way how it works, or you can build up, is that you have these textbook-like signaling pathways where you have ligands, receptors, different mediators, transcription factors, and they are connected to each other. So there are no single signaling pathway that you see in a textbook. They are all intertwined. And the transcription factors in the nucleus are also having different types of complexes, again, very complex. Then they bind to the DNA and regulate the expression of either the genes responsible for the signaling proteins, or, for example, microRNAs that can regulate the translation. And then these microRNAs can indeed affect that. So it's quite complex, and this is a very simplified picture of what is actually happening in a signaling network. And different omics approaches are providing really cool data for the different networks. And as you can see, for the different layers or different networks here, you need to detect different types of interactions, which is often challenging when you would like to integrate them. There are lots of really good databases which store information about these networks. That's good, but also bad, because there are lots of resources that contains different elements of this complex network. So that was a motivation a little more than 10 years ago to create an integrated network resource, which was built on my PhD work, which was this first version of the signaling resource, which was a manual curation of lots of signaling pathways. And here we integrated lots of different interaction and regulatory network resources to get a more unified picture of what is happening in a cell. And this is a screenshot of the download page of this website, where you can search for the species, or sorry, select the species. We have data on C. elegans, Drosophila, zebrafish, and human. For the model organism, this resource has the highest number of signaling data compared to other resources. And then you can select which pathway you are interested in, and then there's a very interesting onion-like structure that helps you to navigate easily on what data you can download. So if you're interested about the hedgehog pathway and only the transcription regulators and microRNAs of that, you select only those two layers, and then only those transcription factors will be downloaded that are regulating the hedgehog pathway. And you can access it in various file formats for modeling, visualization, or just data analytical purposes, or you can just download the whole thing in an SQL. And you can look at individual signaling proteins. So if you are looking at a unique protein page, there will be a link to signaling, and you can look at it as well as from species-specific resources. Lots of people luckily using this resource nowadays. And earlier this year, we published a new version of that, which contains now link RNAs and a very good feature that you can filter the interactions for tissue specificity and subcellular specificity. So if you're interested about only signaling interactions that can happen in the liver or in the gut column, you can get that. We doubled the number of pathways we have. We had more developmental and differentiation pathways. Now we have immune pathways as well. And you can download even data for a single protein, and you don't need to download for a full pathway. So there are lots of new features. If you are doing modeling, for example, there are more data in this new version. And on the right side of the screen, there's a feedback tab. So if you try it, which I hope that you will, and if you experience something weird or something is not there where it should be, just send a quick message there. I'll be grateful for that. So this is about signaling networks, but obviously there are many other types of networks. And with the different omics data sets, actually you can see that networks can be a really nice framework to organize the data and to interpret it. And that's what we often do in our work, and that's why I really like networks, that you can get an understanding of how different layers, like if you add a metabolite or a drug compound to the system, how it is affecting different signaling pathways, and then those signaling pathways, how they are changing the regulatory network. And this is what we are recording practically in transcriptomic profiles. So I think these network approaches can be very useful. And in general, what we would like to do, obviously, is to go from genotype to phenotype. And for that, these type of omics approaches that are now very much available are quite relevant. With machine learning and statistical approaches, we can process the data. Networks can help, as I mentioned, to interpret it. And then ideally, we will be able to do different comparisons between treatments or between healthy and disease conditions. However, it's quite challenging. So there are multiple things. I just listed a few of them, which is, I think, quite clear that needs to be done in order to have a really nice pipeline. It should be scalable, reusable, because you don't want to do it in every project. You really would like to integrate multi-omics and microbiome data, ideally genotype data if you are talking about patients. And often you have a really big data set and you would like to have a proper system for that. So when we collected all these, we just decided that, okay, that's indeed a challenge currently for the field. So let's try to solve these. And one by one, we developed in the last 5 years different tools to tackle these issues. So I'm not going to talk about how we store the data. That's a separate, more informatics-based approach. But we have a really nice tool called Sherlock for that. But we have these multi-layer network resources. I already mentioned signaling, and there are other ones for autophagy, oxidative stress, and cytokine response that contains this type of data. And there are obviously many other network resources published by others. So we created a benchmark called OmniPath 6 years ago, which contains more than 50 different network resources in a one-stop shop approach. And recently, we upgraded this OmniPath approach by adding data not only from human, but also by homology-based modeling from mouse and rats, and also cell-cell interactions, drug interaction data. Now it has more than 120 different resources, and you can access it from R, Python, or directly in Cytoscape if you would like to visualize it. So there are multiple ways to work with the data. Okay, you can just download it on the website. The way how our group works with this type of data is that we create different pipelines. One of them is iSNP, a precision medicine pipeline I'm going to mention today. Another one is to process single cell and bulk transcriptomics data. And the third one, which I'm also going to mention, is microbiomics to look at host-microbe interactions. So basically, we have these pipelines to process data for different experiments. So the key focus in our group is inflammatory bowel disease. And this is a disease that I'm going to present in the next slide, which is quite complex and chronic, but also a disease where systems medicine type of approaches could be very, very useful. So probably many of you have heard this before, but just as a summary, we don't really know the pathogenesis of IBD. It's really a combination of some kind of genetic susceptibility, immune dysregulation, and environmental, probably microbial changes. There are two types, Crohn's disease and ulcerative colitis. And an increasing number of people have IBD now basically globally. And it's quite detrimental for the family and for work. For example, around 20% of IBD patients are not really able to work. The main issue from a clinical point of view is that there are no real biomarkers that can help to suggest which drugs the clinicians should give to the patients, although there are more and more really good drugs on the market. So that's one of the key issues, that how we can identify ways to match drugs with patients. The other one is basically how we can keep them healthy, because in many cases, patients, 70% of the cases, unfortunately, they relapse, they get the inflammation again. So the way how it looks in the gut is basically that in a normal colon, for example, you have different nice cell types there in an absolutely uninflamed situation with a nice mucus layer that keeps the luminal content and certain pathogens far from the single epithelial layer here. While in an inflammation situation, in ulcerative colitis, you have much less mucus, you have leaks, you have inflammatory immune cells infiltrating to the mucosal layer. The key thing is that there's a major change in cell-cell and cell microbe interactions in IBD. And even if we are able to decrease the inflammation here, there should be some kind of regeneration that is happening, and that's one of the biological focus of my group, to understand how we can facilitate that. In terms of big data and IBD, that's quite a fascinating field nowadays. There are lots of data sources that can help to understand that. And in this big review, we summarized many approaches there and analytical approaches as well, basically to help on patient profiling, identifying new biomarkers. What I'm going to cover is how the different high throughput omics approaches and systems biology can help with IBD. And this is where we are going to the more network medicine type of area, which is when we are using these network approaches I presented at the beginning to understand how we can treat the system better. And yeah, it's like a 15-year-old story, network medicine. And you can say it like a think globally, act locally type of approach. Nowadays, there are more and more ways how network medicine is being used, not only for biomarker discovery, but also for drug development and drug repurposing and patient stratification, as I'm going to mention. There was a big hype with network medicine. There were a few very nice papers around 10 years ago, and now slowly it's getting there. The key thing is that, as for every computational approach, you need really good data. And for networks, what you really need is different types of data. So this kind of multi-layered network approaches and then this combination of different types of omics data sets can actually help these network approaches to work properly. So here I just would like to showcase how the network resources I presented and we developed can be used and have been used for understanding IBD-related questions. So as I mentioned, we can have a nice signaling or molecular interaction network in a cell, and we can add transcriptional regulatory circuits there as well. One of the proteins that are quite interested in IBD, in Crohn's disease in particular, is ATG16, which is a key protein in the process of autophagy. And as I mentioned, we have a specific resource where all the autophagy-related proteins can be investigated, and all their interactions and the regulation is there. And we did a study when we removed the ATG16 from the intestinal cells of a mouse. We did a proteomics and transcriptomics approach on these cells, and then basically we were able to point out how autophagy is relevant for different cellular processes. With signaling, we have a really large collection of different signaling and regulatory interactions, but in general, the resource does not have any cell type specificity. What we did in this other project is that we did a transcriptomic profile of, actually they were organoids, which I'm going to mention later, but the point is that very specific intestinal cell types that are relevant in IBD, like goblet cells or Paneth cells. And we combined this omics data, what was present in those cells and what was not, with a general signaling network, and we were able to create the first regulatory network for these cell types, and we were able to point out that there are changes known in IBD that are actually affecting these very specific cells. Then obviously these cells are not alone. Cytokines produced by other cells are affecting that, and with the cytokine link, you can map these. And very recently with Nick Bowell, we had a joint project where we looked at how different cytokines have different effect. Basically, we did a kind of a footprint story for different cytokines, what changes they are doing, especially for those cytokines that are quite prevalent in inflammatory bowel disease. And the other external stimuli is microbes and microbial proteins. And with this microbial link approach that I'm going to present soon, we did a study, and then actually this one was done by my former postdoc in another lab, where we looked at metaproteomics data sets and how they can actually contribute to the inflammatory milieu in Crohn's disease. And then obviously the outputs of these cells are communicating with other cells, and this is what you can get through OmniPass. The interesting thing in IBD that there are, as I mentioned, the genetic susceptibility. Each patient have a different set of SNPs, single nucleotide polymorphisms, that slightly affects the regulation of those proteins. And each patient have different combinations that I'm showing in this animation with red. So different parts of their cell is affected. And what we see actually is that usually these are converging on the same pathways. But when we are looking for drug targets, especially patient-specific drug targets, it really matters where the actual perturbation is starting and what is actually affected by that. So we are interested whether these network approaches can help to stratify IBD patients. And if you are interested about network approaches in general or in relation to IBD, last year we published a review to basically summarize all these concepts and approaches. So now I would like to show 3 use cases when we are specifically looking at different types of IBD-related questions. One of them is this very exciting part when you would like to create patient-specific networks. So the main motivation for that, as I mentioned, is that even if we know now those SNPs from genome-wide association studies that are important in IBD or relevant in IBD, they did not really have clinical impact. So one of the reasons for that is that many of them are in non-coding regions of the genome. So it's not that like in many cases in cancer, you have a mutation in a protein, and because of that, it changes its activation. In IBD, many of these are regulatory SNPs, which have mild effect and often really difficult to annotate. And we don't know in which cell type they are actually working. So we created a workflow where we took in the first pilot study, actually, UK IBD genetics data from 340 patients from East Anglia, and profiled them individually, looked for where the SNPs are, and we found that actually many of the SNPs are in transcription factor binding site locations, or in the mRNA or microRNA target sites, which points out that these SNPs are kind of affecting indeed the regulation of very specific genes. So the way how it works is that on the right side, you can see that we look at the a transcription factor whose binding to the DNA is affected by the SNP. It can be a gain or a loss of function, by the way, or a microRNA that can or cannot regulate an mRNA. And then by that, we can identify the actual protein that is affected. Many of the projects that looked into this stopped there. What we did was that we went literally one step further and looked at protein-protein interactions or signaling networks, and looked at the context of where these affected proteins are. And that was a very key step, actually, on understanding what is happening. So this is a summarized view of one of the projects we did. When you see the transcription factors and the microRNAs here, it's the same arrangement, by the way, that I showed in the previous small network. These are the proteins that kind of their regulation is affected, and these are their first neighbors. And if we do a functional analysis on this, then we start seeing a very interesting thing. You can get very specific functions like calcium homeostasis, antigen presentation, stress regulation, very relevant in IBD, that they are getting affected through these first neighbors. If we would do a functional analysis just on the SNP-affected proteins, you can get some of these functions, but they are not key proteins there. They have other functions. So basically, adding the first neighbors was very key to point this out. What we were interested is that can we use this to understand how patients can be put into different clusters or groups. So if we put everything into a single network, like an omnipass network with all the SNPs highlighted with yellow, including the first neighbors, we get this super ugly hairball, which I hate. These hairballs are not network biology representations. Sometimes you can see them in papers. That's not really useful. It's like a picture from Star Wars after blasting a planet. What you can do, on the other hand, is that if you have patient-specific data, then you can create patient-specific networks. So in this case, we filter the whole network to a single patient, where the yellow is the color codes for those that in that particular patient, these genes are affected, or their first neighbors are kind of affected. The blue shows those that are not affected in this particular patient, but in our cohort, in our IBD cohort, there is at least one patient where those would be affected. So we can basically compare the patients if we plot these networks. I'm not going to show you the 342, but some of them. And when we started plotting it, and then obviously with some machine learning clustering, it was confirmed, we realized that actually you can find really nice patterns and profiles here, which points out key genes that are driving the clusters. So we were able to identify a set of patients where you have this PKCB and PTMNN1 proteins and modules affected. In these patients, it's more on the right side because the PKCB part is negative and not affected. Here is the other way around. And then we had a very strange cluster 4, where it was more very specific parts of the network affected. And we are looking actually now some of these patients, because it's quite interesting to understand how the process works. And there are many other applications of this that we currently were working on, and I don't have time today to present. But it's a really new approach to interpret what is happening in IBD patients, and how we can use that. The key summary from this is that many of these SNPs have regulatory effects, and that's why it's important to look at transcriptomics and epigenetics data. And it's not an individual SNP that will be used for a biomarker, but basically a person-specific set, because that's basically, I think, what defines what is the affected network, what are the affected processes. And the other very interesting aspect that we realized by working on this project, that if you do a randomized clinical trial, you can often fail a drug, because if you have lots of patients there who has different types of genetic modifications, you are missing those specific ones who could respond to the drug treatment. And in our paper, we pointed out a very nice case where we showed that actually 12% of the patients could be susceptible to work with the drug, while the others are not. And this could have been easily missed in a randomized trial. Clinical metadata is quite important, so we are working with clinicians to do this validation. This is where we are now. We have now a really nice pipeline. We are now looking at it, how it's actually working in real life. And also adding microbiome data is quite an interesting approach for us. And this is basically quite important because these microbes are in the gut, are affecting how our cells are working. So we have basically this microbiomics approach, which is currently it has two parts. One we are still working on, but about to finish, is a nice machine learning based approach together with benevolent AI to identify key features that can predict severity or the flares in IBD. The other side has already been published in this paper, but we are still improving it further. Here, the point is that in this one, we are just focusing on proteins. We would like to upgrade it to metabolites as well. But currently, it's just microbial proteins, and we would like to connect them with host proteins and investigate what's the downstream effect of these host-microbe interaction could be. Especially in this project, we looked at autophagy, but we looked at other processes together with Unilever. If you are interested about host-microbe interaction modeling, there is this review on Frontiers in Microbiology, where we summarize lots of tools that is currently available for that. So in this particular project, we were interested about looking at healthy and IBD-associated microbiome. So many of the normal microbiome, as you know, have a really nice anti-inflammatory effect and contribute to intestinal homeostasis. In IBD, there's a dysbiosis, a change in microbiome, and it's often leads to inflammation. We know there's a kind of a list of what microbes are missing, what microbes are present, and we are interested at whether we can identify some of these as key contributors either to pro-homeostatic effect or a pro-inflammatory effect. And we looked at a specific example, Bacteroides tetratheomicron, or BT for short, which is a commensal bacteria, but mostly associated to a healthy state. And we know that it does not induce inflammatory response. In fact, in preclinical models, it has a really beneficial effect. And you have less of that in patients with ulcerative colitis. So there is definitely something that this BT can do to promote health. We were interested at whether we can identify that in a healthy state and what this anti-inflammatory response could be, or if we look at it, what is happening in UC, whether we can get it as a probiotic, for example, we give it as a probiotic, what the outcome would be for that. And then we created a very versatile and very exciting workflow when we looked at both single cell and bark and RNA-seq data sets, but here I would like to focus more on the single cell one, because we wanted to do a cell type specific host-microbe interaction network. So basically using OmniPath, in this case, we did not create patient-specific, but cell type specific networks. So we used a big column single cell, published single cell study, and we created various cell type specific networks and reconstructed from that a couple of interesting pathways like the toll-like receptor pathway. From the BT, they are very known to produce bacterial extracellular vesicles, and they put different proteins into these, and we know that these vesicles can go into the host cells from the literature and then from collaborators. We did a proteomic study on the secreted vesicles. So we identified the proteins, what the bacteria is actually producing. And then basically we identified the host and the bacterial side, and using the microbiome pipeline, we connected the two. What would happen, or what is happening, when these vesicles are going into specific cells? And then basically we looked at how they can affect the different pathways. So very briefly, because there were lots of data in this project, this is a toll-like receptor pathway with the different receptors and then transcription factors at the end. If you look at an upstream part, this is kind of how it looks like with the TLR4/2 receptors. And this is kind of the network representation of the same textbook figure. And this black edge around the nodes represents proteins that can be targeted by Bt proteins. So we found a kind of an enrichment in the toll-like receptor pathway, especially in the upstream part, which is kind of a target for these bacterial extracellular vesicle derived proteins. And then the interesting thing was that when we looked at it from different healthy and disease patients in a cell type specific way, we found really nice differences. One of them that I'm showing here is that looking at normal dendritic cells, you can see that basically the pathway is active. You have all the relevant proteins there. And TRAP is a major adapter that directs the signaling flow, is targeted by one of these Bt proteins. In a UC setup, the pathway is not really active in these cells. Even if some downstream components active, TRAP and others are not there. So basically, we pointed out that if we are going to give this bacteria for as a probiotic to keep patients in remission, then it's very important to understand what stage they are, because some of these proteins would not be good actually to keep patients, to put patients in remission. On the other hand, when they already achieve that, then for example, these bugs could be very useful probably for that. But if you are interested, there are lots of other things in the paper on this. And very briefly, I just wanted to mention what you can do with when you have these pipelines. We were working on that 2 years ago, and then we just reutilized it very quickly to a pipeline to look at SARS-CoV-2 related proteins, and basically did a very quick study, basically paper to provide the whole pipeline in an easily accessible way when you can look at viral proteins in this case and use a transcriptomic output as an endpoint and connect the two. So it's basically a very nice process where where you can actually identify how a viral infection can contribute to differential expression of genes in an infected cell. And the last example I wanted to show is when, again, we are looking at cell type specific networks. But in this case, it's a very complex situation, an epithelial immune interaction in the gut. And it's still a SARS-CoV-2 related project. We were interested about what is happening in the gut, because there were not a lot of studies with SARS-CoV-2 about gut infections. Obviously, everybody was focusing on lung and airway related infections. But for SARS-CoV-1 and MERS, there have been data showing that those viral proteins can have a very major effect through the gut. And we were interested that what if this gut-related, maybe secondary infection can contribute to the cytokine storm that could make COVID quite deadly. So for that, we were interested that how infected epithelial cells, which was by then known from the literature that they can infect intestinal epithelial cells, can talk to immune cells in the gut, and those immune cells can contribute to this kind of systemic effect. So basically, we need to create quite a big connection, starting from the viral proteins, identifying human receptors, signaling networks, transcription factors, differentially expressed genes, as before, and then connecting the differentially expressed ligands from these epithelial cells to the receptors on resident immune cells. And basically, with collaborators and with published data, we were able to collect the relevant information to add to this pipeline. And this is the whole structure of this workflow or pipeline. You don't have to understand the whole point, and I'm not going to present the whole thing. The key thing is that you can have different input data. One of them are a priori knowledge from Omnipath, cell-cell interaction data, and other. And the other is very specific, like single-cell transcriptomics data or proteomics data if you have. And this is the versatility of the platform that actually we repeated it later on with a completely other tissue as well. One of the reviewers was interested whether it can work in the lung as well, and it worked beautifully just by changing the input data. Everything else remained the same. And basically, the network reconstruction is happening in different bits, and you don't need to do anything. It's plug and play. And the output is this integrated network. And then you even get different analytical tools at the end, which helps you to visualize it and analyze it and look at what kind of functions are enriched. I'm not going to talk about the details. It's one of the summary figures from a network point of view, where we pointed out SARS-CoV-related molecules and what pathways in epithelial cells were affected by these, how they change the expression of ligands up or down in these epithelial cells, and what receptors or immune cells were affected by these changed ligands. And we did it for colon and ileal infections as well, which was quite a comparative study. But the point is that at the end, we were able to create this very nice summary figure, which showed that actually there's a few signaling pathways that are very relevant in infected epithelial cells. And through a few transcription factors, you have more or less of certain ligands that are very relevant with T cell and dendritic cell regulation. And then actually we shortlisted and analyzed these ligands to point out that many of them have been known to be relevant in generating cytokine storm, or have been found during the publication. Actually, there was a really nice study that showed that in genome-wide association studies, that those people who are more susceptible to have a severe COVID, they have a higher expression of certain ligands that we identify through this approach. So it was really nicely confirming that this is a good way to identify pathologically relevant cell-cell interactions. Okay, and just for the last few minutes, I just wanted to talk about the BRC organoid facility that I'm co-leading here at Imperial. This is still relevant for networks. And this is relevant because I mentioned to you in multiple projects that we need to have really good data. We need to have very cell type specific data, patient specific data, and then we can get these from organoids. But also when we do these network models, this is basically kind of a prediction. It's a fun thing to do, but in order to have a translational impact, you need to validate that. And unfortunately, nowadays, more and more journals are expecting that you are coming with a really cool validation project. So the organoid facility can address both. So for those who haven't really met with organoids, I just briefly wanted to mention that there are two ways to create these very interesting models. We can create them from human. That's the main advantage of that. We don't need to work with animal, with mouse, for example. You can take a biopsy or you can take an embryonic cell. And then from this biopsy or blood, you can grow organoids either directly from the source tissue, or you can create induced pluripotent stem cells and then differentiate them to nearly any kind of cells that you have. And then basically in a dish, you can start growing them. If it's an adult biopsy-derived organoid, then usually you just get epithelial cells, so like the gut epithelial layer, for example, if it's the gut. In case of iPSC, you can have some other mesenchymal or other cell types, or you can parallel create that. It's a really cool approach. It's quite expensive, but it's really powerful because you can do it from patients. You can keep the genetic background of the patient, which is very interesting. And also you can manipulate the system very, very nicely. In our studies, it's quite relevant because, as I mentioned, the intestinal epithelial layer is quite important, for example, in IBD. And there are many cell types in the epithelial layer that you can't have in cell culture. And even if you have them in mouse, the murine microbiome is very different compared to ours. So actually studying host-microbe interactions in the mouse is not always useful. You take the biopsy and basically in 10 days, you can get these really nice organoids. And this is how schematically they look like. The luminal side is inside and the basal side will be outside, which means that you can easily test cell molecules and potentially later on co-cultures with immune cells, for example. Another very nice approach is to flip it, and you can have an apical out organoid where everything is just the other way around. And this is what we are using to study cell microbe interactions. So it's a really nice way to study cell and cell microbe interactions. And we are also working on a really nice reporter system. We are integrating a fluorescence reporter to the organoids, and then we can screen for that. As I mentioned, we are quite interested about how autophagy is regulated. So we integrate an autophagy reporter, and then we can select those microbial compounds that are affecting autophagy. And this is one of the key aspects of organoid-based screening, where we would like to get to. One option is that you would like to test many different compounds or molecules on the same set of organoids. So you have a few very nicely tested organoids, and then you are looking at the effect of these. And these microbiomics and other pipelines we have can support the outcome of that. And the other one is this one-to-many, when basically you already selected a very interesting compound and you are looking for the patients where it could work. And this precision medicine angle is something that's already working in colon cancer. So they are taking a sample from certain patients and they are testing different chemotherapy agents. And based on the outcome of that, within a few weeks, they can tell the clinician which chemotherapy would work on which patient. And more diseases are going on this direction. With IBD, we are far from this, but this will be a really, really interesting precision medicine tool very soon that we will be able to use. So basically, the idea with the BRC organoid facility is to have this multidisciplinary facility which can provide really from patients to publication type of pipelines, combining experimental approaches with computational data analysis, and also developing very relevant technologies for improved treatment and disease outcome. And there are already lots of existing networks and systems in Imperial that the facility will work. And if you are interested in the next year, there will be a series of workshops to look at different aspects of working with organoids. The key thing, one side is that you can have different organoid models, so low complexity, high throughput one for screening, as I mentioned, but also there are some really complex organoid models that I didn't present, which have low throughput, but you can get lots of really relevant data from that. What is relevant to my talk is that we need computational frameworks where you can get parallel multi-omics readouts from these organoids, where with machine learning and natural biology data, you can interpret the outcome and aid precision medicine and being it reusable. Even if we are doing a gut organoid-based project, I would like to see the whole computational pipeline working for a lung organoid project as well. And as I mentioned, you can test various things: internal molecules, cytokines, microbial metabolized drugs, and there are a couple of projects already, but this is something I would like to be in 5 years' time when we have multiple projects when we are looking at organ communications, gut liver, gut lung axis, for example. So in summary, the way our group looks at it is that there are multiple systems biology options to help personalized microbial studies, from patient-derived organoids or from mouse work, or even mouse-derived organoids. We used to do some of these before, and create very complex organoid systems or screens for a higher throughput testing of how microbial products are working. And then we already established different omics approaches to get lots of data out on these omics systems. And we are currently working on a high content imaging based filtering of these organoids to decrease the cost of these experiments. And with these multi-layered networks, we will be able to create very specific models, how certain microbial molecules are affecting and which cell types. And with patient-specific mutation data, we can move the whole thing to a patient level and point out that in a particular patient, in one of the candidate microbial therapy, can that have a beneficial effect to prevent or the onset of IBD or relapse. And with this, I would like to finish. There have been lots of people in the group in the last couple of years who contributed to these works. I highlighted some of the first authors of the papers that I referred to. And also there are lots of collaborators back in Norwich and more in London and elsewhere who are providing data and helping us to work with these. And I would like to finish. Yeah, that's a group picture. And I also would like to highlight that all the codes that we develop are available in GitHub, so you can access everything. And if you have any questions, that's my email address. Thank you.